Okay, so yeah, as the talk suggests, as the title suggested, as Simon suggested, I'm going to talk about our work in the last few years dealing with, I should probably say, the sources of variability in the neuronal effects of transcranial electrical stimulation, since that's going to be the focus of the talk. And before I get into it, I'll mention that this is really the work of Matt Krause, who's kind of a polymath genius postdoc who's been in my lab for a few years, who really got all this off the ground starting about 5 years ago in my lab, as well as Pedro Vieira, who's a more recent postdoc to join the lab in the last few years. They really did all the work, but I'm just going to talk about it. You know, I think I don't have to explain to this crowd about the long-standing interest in transcranial stimulation of one kind or another. 2,000 years ago, people were putting electric fish on their head to try to cure headaches. And in the 19th century, you can find devices that were made probably for curing headaches as well in the home, and patents for hats that you could wear. And this continues to the present day. So it's kind of a source of fascination for people both inside and outside the field. And in the last 10, 15 years, there's been this explosion of enthusiasm for the method, including at least 500 or probably much more now clinical trials in the works to explore the idea that TES could be a treatment for just about anything and everything. And this list kind of highlights both the exuberance that there is around the technique and also maybe the unreasonable optimism that people have about the range of things that it could treat. And so when you have unreasonable optimism, you have skepticism, and quite reasonably so. So the problem that has arisen, particularly in the last few years, is that when you look at the full range of results in the behavioral literature, there tends to be quite a lot of variability. And this is a very well-known kind of meta-analysis of behavioral studies, but anyone who's kind of experimented with transcranial stimulation knows that there's a lot of variability. So this is, of course, a cause for concern. If we expect to have this end up in the clinic, we want to know that it works and for whom it's going to work, and we're not anywhere near there yet. And around the same time, the question arose as to whether we're actually getting any current into the brain, as Miles just explained very nicely. What really caught people's attention, I think, around this question was a kind of gross study that was done by Buzaki's lab, where they implanted electrodes into the head of a cadaver and tried to measure the electric field inside the skull of a human during transcranial stimulation. And the news they initially reported was kind of bad in the sense that the electric field ended up being very weak, and arguably too weak to do much in terms of entraining neural activity. And of course, this guy is still dead, which means that TES is not good for everything. So this is sort of where we got involved in this as a research topic in our lab. And we were sort of interested in the million-dollar question, which is whether brain activity, as you know, to the extent that it can be manipulated by transcranial stimulation, could ultimately have an effect on behavior. And this is, of course, where all the glory is in answering this question, but it logically has to build on a couple of other questions. And one of them is whether the electric fields that actually enter the brain can influence brain activity. You have to know that before you can even speculate about how brain activity might influence behavior. And before you can ask that question, you have to ask whether the stimulation that you're applying to the scalp can generate an electric field that is sufficient to do anything interesting in the brain. So although these questions sort of here on the left are not as glamorous, they are kind of logically necessary for the rest to work. And so this, although a lot of this was already covered by Miles, I'm going to go over some of our work in this area as well as some other work in the field. We do have some work on behavior in the monkey, which I'm not going to talk about here. Some of it's already been published. So if you want to know about that, I can refer you to some papers. Okay, so the first question I'll talk about is to do with electric fields. And again, like building on this paper from Buzaki, he sort of argued that at the typical intensities you would use in a typical human experiment, the field strength that you can expect to measure in the brain is quite a bit less than 1 volt per meter, often on the order of 0.3 or 0.5 volts per meter. And with a little calculation from people like Maram Bixen's lab, you end up saying that this is about sufficient to alter the membrane potential of one neuron by quite a bit less than 1 millivolt. And here we're thinking about a neuron that just kind of exists in isolation. And if you remember your introductory neuroscience, it's about 20 times that much is required to trigger even 1 action potential and going from the resting potential to the action potential threshold. So if this is correct, then it sort of follows that at least in the simplest model, there's not much chance of generating action potentials directly from the stimulation itself. And so this leads to the kind of existential question of what are we actually doing when we do this? Our approach to answering this was to look at what happens in monkeys. Monkeys, like humans, are primates, and they have a lot of the same features to their brains and skulls. They have thick skulls, they have gyrencephalic brains, so lots of folds and bumps, and they're capable of being trained to do behaviors which are reasonable proxies for the kinds of things that humans can do. Of course, it's not a human brain, but it's probably the best we can do in terms of an experimental model. So we were working with macaques, and to try to get a reasonable approximation of a human TES experiment, we used an off-the-shelf system, the StarStim 8, with the kind of standard stuff that you would use in a human lab, high-definition electrodes, 1 centimeter attached to the scalp, so we're stimulating right through the skin. And of course, the monkey's not wearing the cap, but he has the electrodes on his scalp. And in order to really give this a chance, we tried to really very thoroughly figure out where the current was going during the stimulation, and that entailed getting an MRI for each individual animal. And with the help of Abhishek Datta, who runs Soterix medical company, we were able to generate finite element models that would segment these MRIs into different tissue types: skin, bone, CSF, etc. And since there's a different conductivity associated with each one, you can build a model of the animal's individual anatomy and ask for a given stimulation montage where the current actually goes. And once you've solved that problem, you can solve it in reverse and say, if you wanted to target the stimulation to a particular brain region, where would you need to place the electrodes on the scalp? And that's what we did. And this allowed us to target several different regions in the brain. We looked at prefrontal cortex, visual cortex, and in the work I'm going to talk about today, we were going deep into the brain, the hippocampus, and the basal ganglia. What the model produces is something like this. So this is an example of an electrode montage that you could use. It gives you an estimate of the field strength everywhere in the brain. And in the model that we tested and published in 2016, the prediction was quite similar to what was found for the cadaver, which is something on the order of 0.3 to 0.6 volts per meter. So in our hippocampal recordings, we did our best to maximize the field strength. So we chose a montage that would really generate a strong field in the hippocampus, and then went in with an electrode, with a 32-channel electrode, and recorded from the hippocampus, as well as the nearby basal ganglia, and found at least that the model was quite accurate. So it had predicted something in the order of 0.29 volts per meter, and the measured field strength was about that, give or take about 10%. So that's to say that the field strength is not very strong, but nevertheless in the range of something that might be expected based on the previous work that Miles mentioned to have an effect. So with the field strength at that level in the hippocampus, we started recording from neurons. And the experiment that Matt Krause did, that we published last year, was simply having the animal really do nothing except fixate a spot on a computer monitor. And the idea here was to get long stretches of recordings that weren't interrupted by specific tasks like reaching or eye movements and so forth. So the animal's just kind of staring straight ahead, and we're interleaving blocks of stimulation at different frequencies that would kind of ramp on, and the stimulation would go for 5 minutes, and then it would ramp down. And in between blocks, the animal would get a break of about 5 minutes, and we would interleave real TACS stimulation with sham, and in the sham, you ramp the stimulation on, then you just immediately ramp it back off over the course of about 10 seconds. And then there's a 5-minute stretch where there's nothing happening. And then to kind of simulate the sensation of TACS, you do the same thing again, you ramp it on, you ramp it off. The theory here is that people, or presumably monkeys, are really very sensitive to the onset and the offset of the stimulation, but not as much in the middle. With the monkey, you can't really ask them, although I'll explain a bit how we attempted to assess how effective this sham was later. But I think there's probably a good chance that the animals could detect the difference, and this is always a problem in these kinds of experiments. But this is probably the best we can do. So we had the stim condition, we had the sham condition, and we looked at different frequencies, 5, 10, 20, and 40 hertz. And recorded from the hippocampus. And I will show you one example neuron that we found. This is a sort of slice of the action potentials being fired at various points in time during the recordings. And what I'm showing you here is the sham condition. And you can see that the neuron is kind of firing at random with no particular structure to it. If you just look at the amount of action potentials being fired, it's around 10 spikes per second. And if you look at the phase of a hypothetical kind of 20 hertz oscillation, there's no particular preference for a phase. So it's firing with no temporal structure at about 10 hertz. When you turn on the stimulator at 20 hertz, things change very clearly. So you can see that now this neuron tends to prefer to fire spikes at very specific phases of the oscillation, which you can see here, even though the overall firing rate really hasn't changed, just as you saw in the last talk. So we're not having an effect on the firing rate. There's a very clear entrainment to a particular phase, which is here about 0. And we quantify that with the phase locking value, which runs from 0 to 1. This, as is often the case in this kind of data, when I say this is an example cell, I mean this is the best cell. It was very well entrained by the stimulation. Something unusual about it was that it tended to fire on the sort of down phase of the TACS oscillation, which, as you'll see in a bit, is somewhat unusual. In any case, this is a good example of how a neuron could be entrained to TACS. A more typical example is this one. This one, you have to look a little more carefully to see that it's actually being entrained during the stimulation at a phase of about 90 degrees, which makes more sense because that's the depolarizing phase of the oscillation. And again, no effect on spike rate. The phase locking value here is closer to 0.15, which is more typical. And here's an example of a neuron that wasn't entrained at all. It continued to maintain the same spike rate and no particular preference for phase. What's interesting about this pair of neurons is that they were recorded at the same time on the same electrode on neighboring channels, about 100 microns apart. So that already gives you a sense that there's quite a lot of variability, even on a tiny spatial scale within the hippocampus in terms of the effects of entrainment. So across several batches of recordings in the hippocampus and in the basal ganglia, this was the consistent result. In terms of firing rate, there's no effect of TACS at any stimulation frequency that we could find. We were unable to find any case which was really convincingly an example of TACS changing firing rate of any neuron. But the spike timing changed quite a lot, and this is visible here at all the different frequencies, 5, 10, 20, and 40. If you compare sham versus real TACS, you have the result that a lot of neurons go from being really not entrained at all to being reasonably well entrained at the stimulation frequency. And there's a suggestion that this effect was strongest at 20 hertz, although we don't have enough data to say that for sure. So generally speaking, one could say that about a third of the neurons were entrained at any given stimulation frequency. Around the time that we published this, Alex Opitz published, Opitz's lab published something quite similar in the motor cortex, showing also very nicely that as you increase the amplitude of the stimulation, you increase the extent of entrainment in individual neurons. And they also found a similar preference for phase. I think they're using a different convention here than we are, but generally a preference for the depolarizing phase of the stimulation. And as you saw in the last talk, there was a nice dose response effect where, as you might expect, the stronger the actual field was at the location of the stimulation, the more the neurons were entrained. And this is, these are just the neurons that were entrained. There's a large population of neurons that were not entrained by any field strength. So the upshot of this work is that with standard TES amplitudes, you can influence neural activity, at least in terms of timing, but not in terms of rate, as far as anyone can see. But the effects are quite variable across neurons. So we have already one source of variability, which is that some neurons care about TES and some just don't. We were kind of wondering why some don't, and Matt and Pedro have got a sort of an answer in their more recent work, where they looked at different frequencies of stimulation within the same neuron. So in everything I've shown you before, they were doing batches of neurons at 5 hertz, batches at 10 hertz, etc. Here you see examples of individual neurons that were tested at multiple frequencies. And what you can see is that there are, each neuron actually has its own kind of tuning curve. So this is an example of one that likes to be stimulated at 20 hertz, but this is one that would only show entrainment at 10 hertz. And here's one that was most strongly entrained at 5 hertz, and so on and so on. And so if you just sort of plot out the levels of entrainment here as the different colors, and we've ordered the cells from top to bottom in terms of how strongly they were entrained overall, you see quite a range from cells that were very strongly entrained at most frequencies to cells that were not entrained at any frequency. But in between, you find a lot of neurons that are kind of picky. So they will be entrained, say, at 10 hertz, but not necessarily at other frequencies. And so if you were only to test one frequency, you would end up missing quite a lot of the neurons that are actually entrained. So depending on how you want to quantify this, one could say that certainly more than half of the neurons can be entrained by something, and probably more like 3/4, but we're still collecting that data. So this is, of course, a sort of good news, bad news situation. Again, it does mean that you can entrain a lot of neurons in the brain, but as of this moment, we have no way of knowing which ones will be entrained by which frequency. If it's something that depends on the layer in which they reside, or the morphology of the cell, or something else. It's an open question. So at any rate, one can say that you can get a reasonable electric field into the brain, and you can detect an influence of that field on the neural activity. Of course, we would like to say that this causes that, but as you now know very, very well, it's not necessarily so. There's a lot going on in between, including the potential for phosphenes, the potential for tactile stimulation, as well as everything else that lives in the head, the cranial nerves, the lymphatic system, and on and on and on. So, you know, it's very hard to even conceive of an experiment that would test all of these things, but we have made some effort to look at them one by one. Phosphenes were the one we were most concerned about because even informally testing the device on ourselves, you can often kind of easily generate phosphenes, and that would be a very trivial way to get entrainment of neurons, particularly ones that were in the visual pathways. The hippocampus, of course, does get visual input along with lots of other kinds of input. So, to sort of approach this question, we built on a finding from Bart Kruckelberg's lab, which is that the phosphenes that are induced by TACS tend to be strongest for electrodes that are positioned more anteriorly and weakest for ones that are posterior. And this was a concern for us because, for example, in our 20 hertz stimulation case, we found very nice entrainment phase locking at 20 hertz, but the stimulating electrode, one of them was very close to the front of the head, and the recording electrode was in the hippocampus. If it's really true that what matters is the anterior-posterior positioning, then it shouldn't really cost us anything to move this electrode laterally on the skull. One would still maintain the same frontal position, but according to our finite element modeling, it would sort of destroy the electric field in the hippocampus. So we then have a divergent prediction, which is that if it's all phosphenes, then kind of moving the electrode over here and stimulating over here wouldn't matter. If it's really due to a direct effect of the field in the brain, then it should matter a lot. And the answer is that it does matter a lot. So this is kind of an interesting result in the sense that you see this ghost here, this kind of trace, which means there certainly were cells that were entrained even when we move this electrode and maintain this kind of frontal location. But on average, the effect of the phase locking is now buried in the noise, meaning it generally goes away. So another source of variability is that you almost certainly have the potential to induce phosphenes, but I would emphasize for this result, it's probably not the main driver of the entrainment in the hippocampus anyway. And then the one that has been of great interest to all of us and that Miles talked about in some detail was the potential for somatosensory or tactile input, which is of course unavoidable given that you can feel the stimulation on the scalp. There's no question that there's some tactile input reaching the brain pretty much by definition. The question is just for a given experiment, how much does it contribute? And of course, we were interested in the work by Asamoah and Miles and that group. And so we sort of followed their approach to say, what happens if you take this EMLA cream and wait the 30 minutes after putting it on the monkey's scalp and repeating these experiments. And for this, we also tried to estimate a kind of behavioral threshold for the stimulation. You can't ask the monkeys, but you can do the next best thing, which is to measure their behavior. And as you recall, this is a very, very simple task. The monkey just has to basically fixate on the screen. So normally they can do this at pretty much 100% accuracy. They fixate and they get their apple juice. But generally when you turn on the stimulator, what's shown here in orange is that they do tend to look around a bit. They get a little fidgety and their time on task may go down for weak stimulation to about 80% from 100%. So that's a measure of how much they're feeling the stimulation. It's not the most direct measure, but it's a pretty good estimate. And what happens when you apply the EMLA cream is shown here in green, and it just shows that this effective distraction goes away. So the animal's basically on task 100% of the time, excuse me, which is where it should be. When you increase the stimulation to 1 milliamp, plus or minus, again, the animal gets distracted unless you anesthetize the skin, and then he's not distracted. And this is pretty consistent with what you saw in the previous talk. There's a behavioral threshold of about 1 milliamp. Beyond that, the anesthetic certainly helps, but the animal can clearly still feel it. So that means that when there's no anesthetic, he's quite distracted. He starts looking around more than half the time. With the anesthetic, he's at about 60 or 70% on task. So all this is to say that to the extent that the anesthetic is doing its job, and to the extent that the somatosensory input matters, we should see a reduction of entrainment when you put the EMLA on the skin. And this is from a paper we published actually just last month with Pedro as the lead author. And the upshot is that if there's an effect of this anesthesia, it's quite small. So we have our standard effect that I mentioned before, which is TACS has a phase locking value that is on average across all neurons about 0.05 in this sample. When you apply the topical anesthetic, it does go down a bit, although the difference is not significant. And it certainly doesn't go to 0. And that's true at 2 milliamps, and it's true at 1 milliamp as well. So at least in the hippocampus, and at least in these monkeys, there's not much effect of topical anesthetic. I certainly would not want to say that that means that there's no possible effect of any somatosensory input anywhere. I think it has to be evaluated on a kind of case-by-case basis. And in this case, it doesn't seem to be the main driver of the entrainment. So that's that. And then, of course, you know, we could keep going down this path for quite a long time, listing all the things that could conceivably influence entrainment. And I think that's absolutely worth doing. The problem is a lot of them, we don't really know how to think about them, or we don't have good experiments. So all we can surmise is that there's some unknown force which is influencing that would be kind of mediating the effect of TACS on neural activity. And that's very hard to sort of deal with without years and years of testing. The list goes on for quite a while. There is one point that I will just mention. There's two actually in this regard. One is that, as you saw before in the phosphine control, the influences that we're finding in the hippocampus, the entrainment effects, are actually very specific to both the placement of the TACS electrodes and the placement of the recording electrodes. So I showed you that if you move the stimulating electrodes across to the other side of the scalp, the entrainment goes away. So it's not simply the case that kind of stimulating anything in the skin or the cranial nerves anywhere leads to entrainment in the hippocampus. Nor is it the case, as you can see here, if you maintain the same stimulating montage and just move the electrode by a few centimeters, is it the case that the phase locking is maintained? In fact, it goes away completely. So, you know, this effect here is based on a montage that we've calculated very precisely to activate the hippocampus, and deviating from that either by changing the montage or recording from a different location makes the entrainment go away. So, of course, this is not total proof that it's a direct effect, but it's consistent with that idea. And the other thing that's consistent with the direct influence is that we do have this finding that the cells really tend to line up in terms of entrainment quite precisely, for the most part, to the high amplitude phase of the stimulation, meaning when the membrane potential should be going up. And we can guess that a lot of indirect influences would impose some kind of delay between stimulation and the effect on the neurons. If you had a polysynaptic pathway or something else that had to change in the physiology of the animal, this entrainment might occur at a delay, which does not really happen in our data. So here, this is 90 degrees is the very peak of this, this oscillation. And this is 20 hertz stimulation. So even a delay of about 6 milliseconds would have shifted this by 45 degrees. So again, this is not total proof. It's certainly possible to imagine a circuit where you've entrained something in the brain at a phase of 90 degrees, and then that has sort of hijacked the entrainment in the hippocampus. But that, you know, this is a kind of interesting mathematical question because it requires a few things to be true at the same time regarding the magnitude of the entrainment, the timing of the delay between some other area and the hippocampus, as well as a few other factors. So it's a good question for modelers. But anyway, as far as we can tell, the effects that we see in the hippocampus are at least consistent with the direct influence. And I think that's useful to know. It may be something that's specific to the hippocampus. We haven't really gone all over the brain looking for this. I imagine that if you did the same experiment that we did in S1 somatosensory cortex, you would get a very different answer, but we haven't tried that. So, indirect effects are absolutely a source of variability in these kinds of experiments that are to some extent unavoidable, but can be mitigated by a careful choice of a stimulation montage and a brain region that you're targeting. I don't know how much time I have left. I'll mention a few recent effects, findings that we have regarding the neural effects of TACS, and then I'll stop. The one that I think is kind of the most interesting and the most recent, so it's still a bit preliminary, but it has to do with the fact that, we've talked quite a lot about the strength of the electric field at different points in the brain and how that really might matter for these entrainment effects. And one often forgets that an electric field is not a scalar field, it's a vector field. And that means that the current will have at each point a sort of directionality to it, which oscillates as the electric field oscillates. And so in considering the possible entrainment effects as well as their variability, we should take this into account. And in the primate, it's absolutely critical, right? Because the primate brain is notoriously folded and has this interesting structure to it. And what that means is that the cell morphologies vary quite a lot from one point to the next. And what that means is that you would expect the effects of entrainment to vary quite a lot from one point to the next. The general finding, which has been elucidated by Bixen and others, is that what should matter in terms of the effect of the electric field is the alignment between the field and the cell body itself. So when these two things are parallel, you have the potential for a strong effect, and when they're perpendicular, it should be weaker. And what that means in the primate is that you have now a very large source of variability, but also a kind of opportunity to do some targeting of your stimulation effects. So Matt and Pedro have been looking at this in the superior temporal sulcus of our monkeys, and this is a region that includes mostly visual and parietal areas, V4, as well as 7A and so on. And by carefully placing the electrode, you can end up with one electrode array going through parts of the sulcus and the gyrus that contain neurons that should be really very differently oriented in space. So at the top of the gyrus, you have these vertical guys. Further down, you have these horizontal guys. And this is over a space of just a few millimeters. So the actual strength of the field is not varying really at all. All that's varying is the orientation of the neurons. And so by recording these neurons with a single electrode, we can ask how much this really matters. And so Matt and Pedro have been doing this in the context of an electric field that is really running kind of anterior to posterior and back with TACS. And so what that means is that you can make a very clear prediction that here at the top of the sulcus, or I should say the gyrus, you have the field going one way and the cell orientations on average are kind of orthogonal. And when you do that, here we have different stimulation frequencies, 5 hertz, 10 hertz, 20 hertz. And in blue, we have the sham conditions. So this is when there's no real stimulation. And these other two concern real stimulation with slightly different montages, so you can sort of lump them together in your mind. But the upshot is that there's really almost no entrainment effect here. And there's a kind of strange de-entrainment, which I don't understand here, but by and large, there's no consistent entrainment effect, which is in contrast to what happens when you go further down into the sulcus and things should be better aligned, where you find the more familiar entrainment effect, where the cells are really well entrained relative to the sham at all these stimulation frequencies. So this is a nice example where the what matters is not the field strength per se. Of course, the field strength has to be something. It can't be zero, but I mean, you have an effect where what's dominating the ultimate entrainment is the orientation of the cells. Do I have any time left, or should I stop? I've lost track. Oh, someone. If no one tells me to stop, I'll keep going. I have one last thing to mention. It's sort of an example, so I'm just going to mention it very quickly. Something we've been searching for in the lab without a whole lot of success is for an entrainment effect that persists beyond the duration of the stimulation. Generally speaking, when we turn the stimulator off, the entrainment goes away. But we have found in a minority of cells a more lasting entrainment effect, at least on the time scale of minutes. And I'm just going to show you one example just to maybe motivate some thinking or discussion. The way we've done this is to plot scattergrams which capture the timing between individual spikes. So if you measure the time from one spike to the next, and then from that spike to the one after it, you can construct a scattergram where you're basically plotting the timing between triplets of spikes. And you can fill this in for all the spikes that you record, and you can make a nice little picture of it. And what should happen if the neurons are being well entrained is that you should get a blob at a certain point, which corresponds to the stimulation period. So this is just an example to show that this does work. I would not say that you find this in a majority of neurons. It's actually in a small minority, but you do find it pretty consistently in our database. So in the case of sham, and now this is 40 hertz stimulation, so what should happen is that you get a blob around 25 milliseconds, right? And that's what happens during real TACS. You find this very robust entrainment effect where spikes are happening 25 milliseconds apart. If you compare sham and post-sham, there's really no big difference. The spikes are happening whenever they happen. The effect that I want to highlight here is that even 1 minute later, you still see the neurons chugging away with this 25 millisecond delay between spikes, which means that there is a lasting effect. Beyond that, it's been very hard for us to find any effect that persists beyond the duration of stimulation. I think that's something that also contrasts quite a lot with human behavioral work. Okay, so to summarize, the sources of variability in our data at the neuronal level concern the distribution of the electric fields themselves. So even small changes in the way that you're introducing the stimulation can exert large influences on the kind of entrainment that you see at the level of single neurons. And that's, of course, important. And I would suggest that everyone should, in designing their experiments, think about the montage for the individual subject that gives them the targeting that they want. Getting from electric fields to the brain is tricky. There's a lot of stuff in between, and one should not minimize that fact. We tried to look at a few of these possibilities, and I think there are certainly cases where there are not likely to be strong indirect effects, but there's no question that there are cases where there are likely to be strong indirect effects, and that in itself is a source of both variability and confusion in interpretation. Once you've got an electric field in the brain, what matters is the strength of that field, but also the orientation of the field relative to the neuron itself. And for reasons that we don't yet understand, what matters is the frequency for that given neuron. And this will have the net effect that you're at any given frequency stimulating some unknown pattern of neural activity that would change if you happen to select a different frequency. And that's a bit of a problem. And finally, to get to this last thing, which is if you can change activity in the brain, what does it do to behavior? That's a tough one because it's what the field of systems neuroscience has been obsessed with for at least 30 years. And we're not going to solve that problem right away. But I would say that a problem is that in this field, we're used to thinking about the sort of neural code that generates behavior as being really a rate-based code where you change the rate of some neuron's activity or a column or some bold signal, and that co-varies with perception or behavior or something. And it's often very localized in space and kind of averaged over time. Here we have an effect that is not like that at all. So if you're manipulating neural activity with TES, you're probably not changing the rate of anything. You're probably changing the timing of it. And you're not doing it in a localized fashion in a column or in an area. You're doing it in a distributed fashion. And we don't have great models for that in terms of relating that to perception or behavior or other things that we care about. So that's a real challenge for both computational people and experimental people. So I'll stop there and just say, here's Matt trying TACS on himself. And here's Pedro. And as I said, these guys are really the world experts, as far as I can tell, on sort of setting up monkey electrophysiology in combination with TACS. They really did a great job. We had help from people who did modeling, in particular Gavin Cooey, who was an undergrad in my lab and is now a grad student, and Abhishek Data, who got us started on the FEM modeling. Excuse me. So I think I'm over time, so I'll stop there.